Okay, great. So, hi everyone. Thanks Natalie and Christine for letting me talk. I'm really excited to get to share some of my, well, at this point it's research hopes with all of you as I get my lab set up here at McGill. Hopefully you're seeing my, my slide and not the presenter view. Can someone just nod to confirm that? I always struggle with this when I teach. Yeah, okay, good. Okay, so what I want to do today is sort of give a bit of an overview of what my PhD work and what my postdoc work looked like, and then sort of tell you a little bit about how I see this tying into the department here and what my plans for the future are. And I'm just plugging for my new lab group. This is going to be a bit of a promotional event. So we're called the Biofluids and Global Health lab, but I use the royal we here because it's just me right now. So if anyone's interested in reaching out for research positions, I'm very excited to work with people. So the title is Engineering Applications to Global Health from Biological Gels to Population Level. Understand the important role that I think bioengineers and engineers in general play in disease transmission and understanding that. So just to sort of give a context for what we're going to talk about, if we think about global health challenges, which we could say as possibly motivating bioengineering design, biomedical engineering approaches and whatnot, you get a very different picture of what, say, the big challenges are if you look at different places in the world. So this is data from the WHO for 2016. You can see that the top 10 causes of death in high-income countries on the left versus low-income countries, the colors look very different. So the color blue corresponds to non-communicable diseases. So you can see that this is far and away the leading cause of death in high-income countries. So you could think heart disease, stroke, things like that. Whereas low-income countries still have a substantial burden associated with communicable diseases, including respiratory infections, HIV, AIDS, malaria, and others. But it's interesting to note that this was all turned on its head on one day a few months ago, which was April 7th, when the daily death toll from COVID-19 in the US, which is a communicable respiratory infection, exceeded that from the usual leading cause of death, which was heart disease. So I think this picture gives us the idea that in general, global health challenges are different depending on where you look, and we should be motivated by challenges across the globe and not necessarily just where we ourselves are located. But furthermore, communicable diseases still remain an important challenge for people who think about bioengineering and medicine. So there's tons of amazing examples of engineering applications to disease, both communicable and non-communicable. We can think of designing biomaterials and how that's helped in, say, mucosal diseases, diagnostic devices, other devices like stents or prosthetics. We can think about sort of more cellular engineering, like thinking about immunological aspects. We can think about the design of vaccines and therapeutics, which is obviously a very relevant topic right now. And I think maybe, at least to me, when I was going through my engineering job, was made a bit less. And I really think about this as going about from two different mechanisms, and I'll elaborate on these as we move forward. And these are related to how we sort of generally characterize disease within a population. This is particularly for communicable diseases, not non-communicable diseases. We think about individuals sort of largely falling into either a category where you're susceptible to something, or you're currently infected with something, or you're immune to something. So what we can think of is some of the technologies that we developed as engineers could either be targeting how the transmission of disease is occurring in the first place, or what it might be doing is changing the relative fraction of people in each bin. So you can think, for instance, that's the whole point of vaccine design, is you want to move people from the green category of susceptible into the gray category of immune. So these are sort of an introduction to some of the ways that we can manipulate how disease transmission works at the large scale. So to reinforce this point even more, I want to sort of break down this framework, particularly in the context of COVID-19, since why not? That's all we talk about these days anyways, but also that will be the subject of the second half of the talk. So for instance, if we were thinking about how different challenges or questions can be broken down in this framework in terms of susceptibility. We could ask ourselves, well, what fraction of the population is still susceptible to the disease? And the flip side of that is related to the immunity question, and it would be what fraction is immune. So you hear a lot about in the news now, people thinking about this. For instance, one proposed method is to use serological tools to measure things like antibody prevalence in individuals to get an idea of this landscape. Furthermore, if we wanted to think about the immune question a little more, we could ask ourselves, what does immunity even mean for COVID-19? And this isn't something that's clear right now, and that's actually going to be the focus of the second half of the talk in detail. You could also think, well, if we were interested in tuning the sort of infection stage or transmission stage, we could think about the effect of different policies on transmission, such as wearing a mask. Is it useful? Is social distancing useful? And then lastly, for instance, I would think that something like a vaccine or an antiviral would actually play with many of these categories, such as changing transmission and also changing the structure of immune classes. So I think that this is a helpful framework that helps me to think about where my work lands, even though maybe at first glance it seems a little bit like I've jumped around a lot between departments, between my PhD and postdoc, although just a plug to say that that's very much still possible. And if it feels like you've pigeonholed yourself into something, I would say that that's not true, and just do what interests you. It all works out in the end. So to sort of contextualize my work, then I spent a lot of my PhD thinking about the question of susceptibility by focusing on one specific aspect of the human body, which is mucus, and that's a main component of the innate immune system. So really what we were trying to understand was how the protein component of mucus, which are known as mucins, serve as a barrier to infection and protect the body in a number of different ways. And so we are interested in thinking about how the structure of mucus can be altered and what that means in terms of changing host susceptibility to infection. And this is more relevant than ever these days, since SARS-CoV-2 gets in through the airways, which are lined with mucus. And so having a good understanding of what protective role innate immunity is playing there is very important for understanding the infection process. So I'm going to start by introducing some of this work, and it's related to us trying to better understand the physical and chemical properties of mucus gels. Next, what I'm going to do is talk about a problem that's related to the immune section, and that's how the nature of adaptive immune responses to SARS-CoV-2 and a potential vaccine might affect the dynamics of COVID-19 in the long run. So this is work that we just put out recently, and so I'm very excited and motivated to talk about it. And I think that it sort of showcases how the multiple aspects of this framework are connected, but still help us think about transmission at the larger scale. And I would go so far as to argue that a bunch of the other projects I've worked on can also fall into these bins. For instance, I spent some time during my PhD thinking about using mucus as a diagnostic material. And during my postdoc, Natalie read off a very long list. I'm not an expert in all of those things. It was just a lot of these things are used, such as climate or behavior, are used to think about transmission at the scale that we're modeling it. But that is not to claim that I am an expert in those things. But we did do some work thinking about how climate might affect disease transmission. I'll touch on that a little here, but we've done that in the context of other diseases as well. And then some other aspects of within host modeling infections. But today I'm really going to focus on these two projects that I illustrated before, and then I hope at the end to tie them together and convince you how all of these things are related. So first, we're going to talk about mucus. So in particular, we're going to think about how the biochemical structure of mucus sets its mechanical properties, which are essential for function. So mucus is a pretty amazing biological gel. It coats all wet, non-keratinized epithelial surfaces of the body, and that's what you see with mucus man on the left here. And the main structural component of mucus is a large glycoprotein named mucin. And the body produces about a liter of mucus every day, so that's pretty amazing. Now, depending on where mucus is located in the body, it has different properties which allow it to serve different functions. So for instance, the environment knows how to do that by just tuning what's going on around the mucus, and then it has, as a result, it looks kind of different. So for instance, in the eyes, the tear film is a mucosal fluid, I'll call it, but it's very watery and thin, and it prevents you from sort of incurring damage by friction when you blink, among other things. And as you can see from Mucus Man here, that is occurring at a sort of pH neutral environment, and that's allowing the tear film to have the properties that it needs in order to serve the purpose that it does. In contrast, in the stomach, which actually has the same mucin as in the tear film, it's known as MUC5AC, so there's different types of mucins in humans, you have a thick layer of mucus that coats the cell wall inside the stomach and essentially prevents you from ingesting yourself, since you have stomach acid contained within your stomach. So something about the different environment is causing this mucus layer to have very different properties, but it needs to in order to serve this purpose of like staying stuck against the stomach wall and not just sliding off. So as much as the environment can play with what the properties of mucus are, pathogens have figured out that they can do this too. So for instance, one of the big diseases related to mucosal dysfunction is cystic fibrosis. And because of dysregulation of ionic channels in the where the mucins are being secreted, the mucus has very different mechanical properties, which make it difficult to clear and prone to bacterial infection, which can ultimately be deadly in people who have this condition. And then similarly, there's a bacteria known as Helicobacter pylori, which causes stomach ulcers, and it's figured out how to secrete the right sort of chemicals to locally change the pH of mucus in the stomach in order to be able to swim its way through and infect underlying cell layers. So to start to understand the structure in a little more detail, we have to think of what these building blocks are in the mucus. And so as I mentioned, mucin is the main solid component. So it's a really large glycoprotein. And so the structure is such that it has a protein backbone. And then these red domains are sugar chains that stick off, kind of like a bottle brush, if you're familiar with those. And in solution, these aggregate into very long chains through what some people call sort of dynamic covalent bonds end to end. So they're not quite covalent, but they're stronger than a non-covalent bond would be. And then the network also forms through a bunch of other mechanisms because these mucins are quite biochemically complex, including electrostatic repulsion between the sugar chains, which are largely negatively charged, hydrophobic interactions between hydrophobic domains on these molecules. Sorry, and that's it. Sorry, I didn't mean to jump slides there. So our experimental system then was to form reconstituted gels made up of this type of protein as opposed to dealing with native mucus, because we wanted to be able to control what went into the gel to a larger degree than when we just got a native sample, say from the stomach of a pig or something, which is the usual animal system for obtaining these mucins. So we reconstitute gels from the mucins themselves. Everything I'm going to talk about today is MUC5AC, which is a particular mucin I introduced earlier and explained that it is in different parts of the body, but the gels have different properties depending on where they are. And so what we tried to do then was to change what we put into the gel in order to be able to tune these types of interactions in different ways. So for instance, if we change the pH of the gel, then what that does is it changes the charge and the conformation of the polymer backbone. It causes the mucins to either be more rigid or more floppy, which changes the properties of the gel in some pretty complex ways, as we'll see soon. We also did experiments where we added surfactant in order to target hydrophobic interactions. And finally, we did experiments with salt to try to change the degree of electrostatic repulsion. So I'm going to summarize those at the end, but I won't speak about them in detail. I'm just going to go through some of the experiments with pH and then try to wrap this part up and explain to you why we think it was important. So the first experiment that we did, and others had done it before, was to take these gels and to make them at different pH and then stick them in what's known as a rheometer in order to measure their macroscopic rheological properties. So here what you're seeing is, for those not familiar with rheological plots, G prime corresponds to the solid-like component of a gel. So its ability to store energy, and those are the filled circles. And then G double prime corresponds to the liquid-like component of a gel, or its ability to dissipate energy. And so the first thing we saw was that if you treat some polymer physics quite loosely and you interpret these gels as having some sort of a plateau at high frequency, you can interpret that as being a measure of the mesh size of the polymer gel. And so if we, again, sort of hand-wavingly look at this data and say, well, it looks like as we're decreasing the pH, these gels are becoming stiffer because these properties are increasing in magnitude. Our first picture from macroscopic experiments was to guess that what this meant was that the mesh additional connections were forming in the polymer mesh as we were decreasing the pH, causing the network to become stiffer and causing the properties that we measured to increase as we drop the pH. So you can kind of think about it as a bunch of spaghetti noodles. And if you were able to add some twist ties to tie some of the spaghetti together here and there in order to make it look more like a soccer net or a basketball mesh, as opposed to just floppy spaghettis floating around, then they'd be able to, as a network, be able to store more energy and to resist deformation more, and overall it would become stiffer. So that's sort of the picture that you get at the macroscopic scale. So this is going to eventually be a two-dimensional plot, but for now we're just saying that one thing we saw for these gels was that maybe what's happening at different pHs, and this is consistent with what we said before about the stomach lining needing to be quite thick, was that as we are dropping the pH, these gels were becoming stiffer. And we hypothesized, and this has been shown before using a bunch of different techniques, that this might be to do with the polymer molecules becoming stiffer and more elongated and exposing more hydrophobic domains that permit additional crosslinking. So this is quite a large scale picture of things. So for those who don't do macro-rheology regularly, these plates, you're going to be hard pressed to find plates much smaller than about 20 millimeters. So this is quite small. So what we wanted to know was what was really happening at the smaller scale. So particularly for mucins, which we know are interacting with pathogens and with a bunch of other things, if depending on the spacing between the mucin molecules, which is known as the mesh size, this is what I was talking about earlier, if we're thinking about a soccer net that's becoming smaller, having smaller and smaller windows, the mesh is getting smaller and smaller. When we compare that size with pathogens of interest, say viruses or bacteria, we start to realize that the environment that these things might face is going to look quite different to what the rheometer plate is going to face. So you can imagine if you're a small little guy who's able to swim through the mesh, you're basically completely unaware that you're in a gel, and you might just think that you're in a nice solvent environment or water-like environment and really have no idea about what's going on around you. Whereas if you're larger than this mesh size, then you are affected by the structure of the gel and by the solid components. So this is just to summarize that and say that below a critical size, we can think of rheological properties as being dependent on the length scale at which you take them. So to verify this, we did what's known as single particle tracking, which is where we took a 1 micron particle, and it doesn't have any energy. We're not applying energy here. We're just letting it passively diffuse due to random collisions with its environment. And what we saw was that delta average x squared, this is the mean squared displacement of these particles, was actually the greatest in the pH2 gels, which we had measured macroscopically. These were the stiffest, and then was the least in the pH4 gels. So you can imagine this is a little bit counterintuitive, because at the macro scale, we thought that our mesh size was the smallest at pH2, and then microscopically, what we were seeing was that it almost seemed like, you would think, okay, if this is a really stiff environment as a particle, I'd be moving quite slowly, whereas all of a sudden these pH2 gels appear to be able to diffuse much more readily. So the mystery could be resolved by not looking at the ensemble average of all of these trajectories, but looking at the individual trajectories. And what you can see is that at pH 7, you get quite a homogeneous picture. All the particles are doing more or less the same thing. But at pH 2, you get a really different picture. You see that each particle is essentially doing something entirely different. Some of them are very confined and hardly moving, whereas other ones seem to be diffusing almost as if they were just in water. So this was interesting because it means that, yes, the gel is getting stiffer from the point of view of a large scale measurement, but there's something else going on at the small scale that's not just sort of a pure cross-linking stiffening phenomena like we'd be used to if we were thinking about maybe a more simple non-biological polymer. So to try to visualize this a little bit more, we used little reporter peptides, which are positively charged. And with the hypothesis that they are very, very small, so they're not really going to do much to the structure of the gel, but we expect that they will bind to the negatively charged mucins. So the idea was to see if they could almost serve as little tracer particles to show us what the network looked like, because ultimately it's very hard to visualize mucin networks. We can try to infer what they look like from looking at the motion of little tracer particles inside them, but making these mucins fluorescent is not obvious in all scenarios, and visualizing in other ways can be complex, especially if we're talking about at the level of individual molecules. So we thought this could be another approach to try. So the experiment goes as follows. We take these little positively charged peptides and say you just put them in pure buffer at pH 7, you see the following. You see that everything sort of looks homogeneously lit up. It's not a very interesting picture. If we now add mucin, also at pH 7, and the reporter peptides, you see not much has changed. They still look very evenly distributed within the gel. Then when we do this at pH 2, so we just wanted to double check that it wasn't something about just changing the surface charge of these peptides that would make them crash out or behave strangely. So they do seem to do just fine at pH 2. They're still evenly dispersed within the solution. But finally, once we add mucin, what you can see is you now see these really distinct cloudy regions where you have some essentially bare spots, and then other regions where there seems to be really dense aggregation, which we think is related to some sort of mesoscopic phase separation, where you get some regions where these mucins are forming these additional crosslinks and the gel is sort of stiffening from the perspective of what we were talking about earlier, but that comes at the expense of creating these gaps, which is where some of the particles were easily able to slip through. So we did these experiments using the other methods I was talking about as well to target other aspects of the gel formation. And this lets us put together sort of a picture of what we think the sort of knobs are that we can tune, whether, of course, in the body, we don't dump surfactant in, so that's not a key candidate for an in vivo approach, but essentially how we can modulate how the network is formed and what that might do to the properties of the gel. And so we think that this was important because it's interesting, even though mucus is so essential to so many functions, the structures of the molecules themselves still remain quite mysterious because of how many of these sugar chains there are that stick off, which seem almost random in terms of which ones are there and how they are ordered. So there's still a lot of mystery related to the actual structure of what these mucins look like. Throughout my PhD, I must have been asked about 1 million times how many cross-linking sites each mucin molecule has, and the frustrating answer was we just don't know. So it was really hard to do some real sort of estimates from polymer physics and back predict what these properties should have been like from the structure itself. But in any case, by starting to do these experiments and by doing the analysis with the different mathematical tools that we have, we started to get a better picture for how the network forms. And ultimately, moving forward, my goal is to think about how these gels are serving as protecting us from interactions with viruses or pathogens and whatnot. And so understanding their behavior under different conditions will be really important for establishing that. Okay, so that wraps up the first part. I'd like to shift gears a little bit now, but I promise to bring it all home later and explain why this is important. But I thought, since this is an engineering crowd, I might take the liberty of doing a mild introduction to disease modeling before I explain to you how we did the work on COVID-19 that we did. But it's not going to be, I'm not going to try to teach too much, but I just think it might be helpful for explaining some of the results that we're going to get. So as I was saying, I think one thing that struck me during my postdoc was how simple the models are that are used in disease modeling to begin with. Essentially, we treat the world as being split, or our population of interest as being split into three groups. We have people who are susceptible to something, we have people who are currently infected with something, and we have people who are recovered or immune from something. And we say that the total population N is just the sum of all of these people. If we're interested in demographic considerations, we can impose a birth and a death rate, which we denote by mu, and then you just have people flowing in and out of each of these bins at rate mu. So this assumes non-fatal infection, so the birth rate is, so the death rate is uniform across categories here. Then what we can do is define a transmission rate, which is a rate at which people move from the susceptible category to the infected category. And physically, what this transmission rate is the product of the number of contacts an individual makes and the proportion of each contact that gets infected. And then lastly, we define what's known as a recovery rate, which is the inverse of how long you're infected for, and that fixes the rate at which you move from the infected to the recovered class. So for mathematical purposes, it helps to take this n equals to 1. So we just essentially normalize the population, and then we can write out the governing equations where now S, I, and R are not the number of people, but they're the fraction of the population. So the governing equation then for the susceptible class is that the time rate of change of people in the susceptible category is related to the birth rate, because everyone who's born is assumed to go into the susceptible class. There are some subtleties in some diseases where if you have some maternal protection from maternal antibodies, then this might be sort of complicated, but in general, we can just assume that people who are born are initially susceptible to an infection. Then they move out of the susceptible class. So you can see that the rate at which this occurs is the transmission rate times the number of susceptible people and the number of infected people. So both of those matter in terms of determining how many people will become infected. And then people also flow out by dying. And then we can do similar analysis for the infected class and for the recovered class. So I thought I would just plot what this looks like. I just plugged this into MATLAB and I ran an imaginary epidemic where I introduced one case into a fully susceptible population, or a small number of cases into a fully susceptible population. And I just plotted what all these curves look like over a time period of, this is 1 year here. And so what you see is kind of interesting. So first of all, initially everyone is susceptible, no one is infected or recovered. With time, the number of infections increases, but the number of susceptible people also increases. So I like to think about it as sort of a fire where you put wood in a fire and then you light it. Eventually, even if there's lots of coal and fire particles left, that's a horrible analogy for how a fire works, but if there's no more wood left to burn, then the fire is going to die out. Or if you take all the oxygen out of the air, the fire is going to die out. So the number of susceptible people really is important for determining how much the infection can still spread. And that's essentially the whole motivation for vaccines. But what you can see is another interesting feature, because we have included births in this model, and so we're constantly supplying the susceptible class, is first of all, sorry, I didn't mean to do that. You can, not everyone gets infected, so this I peak doesn't go up all the way to 1,000 here. So that's one important feature of SIR models. But then at equilibrium, we maintain a constant number of infections, and that's because we have included demography in this model. So you can ask yourself, well, under what conditions will infection spread? So I obviously chose a specific set of parameters when I made those plots that cause the infection to take off in the first place. So you can easily just show that by showing that the time derivative of I should be positive at time 0, that you get the following condition. And this is what sets the basic reproductive number, or the R0 value that you've all undoubtedly heard countless times in the last 6 months. So essentially the condition is that this number has to be greater than 1 in order for the infection to initially spread. And the technical definition of this is the number of secondary cases that arise from a single infected person in a fully susceptible population. So this seems great, but it's quite simple. So obviously there are lots of factors that might complicate this. So for instance, the transmission rate that I just explained as one value beta may be a function of time. And there are a lot of important examples where this is the case. So for instance, this is a plot from a really nice paper about measles transmission. And what you can see is that when these authors sort of backtracked what the transmission rate would be over time, they saw that the peaks really coincided with when people were in school, kids were in school, and then there was a nice dip at Christmas time when everyone went home, and they even saw like Easter lighting up as another dip because the kids went home. So for these childhood diseases like measles, the timing of schools used to be hugely related to transmission rates. Climate might also change transmission, and this has been another hot topic in relation to COVID-19. So we know that some respiratory infections like flu tend to do better in cold, dry conditions as opposed to humid, wet conditions. Although this is a big mystery because in the Southern Hemisphere, peaks in influenza correspond with the rainy season. So this is definitely still not a resolved question if anyone wants to work on this. And I think that this is actually one place engineers could really contribute, would be to nail down this debate a bit better about what climate does, because if you just do this simple droplet experiment where you have evaporation of a droplet that I've emitted at different temperature or humidity conditions, it's not obvious how this question should be answered. And furthermore, when you actually look at what this debate comes down to in terms of people claiming things like aerosols versus droplets, a lot of it's based on papers from about 100 years ago, and this distinction is really not clear either. And so I think that this is, in particular, one place that engineers could really contribute would be to help helping to understand what role climate should play in the actual transmission process based on very simple arguments related to thermodynamics and fluid mechanics and heat transfer and mass transfer and stuff like that. Otherwise, there's other factors such as demographic realities that can introduce stochastic noise to our system. So for instance, I've presented these models, I call them sort of the ideal gas disease models, where everyone is just a little gas molecule bumping around, hitting other people and infecting them. And this might be appropriate in a large city where you have good mixing, but in small towns, this phenomenon becomes stochastic and you could locally extinguish the disease by having no one infected. So that changes the dynamics quite a bit. And this is a nice plot of measles cases in England and Wales, where you can see some places like London are going to always have cases, but then other places are going to sort of blink in and out, and that's related to stochastic issues from demography. So lastly, I just want to explain a little of the theory behind vaccination. So we can take the model that I showed before, and in the simplest form, we can just say that introducing a vaccination allows us to move people from the susceptible to the recovered class at some rate P at which we vaccinate people. So our governing equations now become as follows. So now you can see they look exactly as before, but I've just facilitated a hop from the S category to the R category without having to go through the I category like you did before. And so it turns out you can use this very simple model to ask yourself, if I want to eradicate a disease, which means I won't have any cases at equilibrium, what's the critical vaccination rate, or what portion of the population do I have to vaccinate? And the result you get is quite a famous one that says that it's 1 minus 1 over the reproductive number. So we can plot what this looks like and then think about where different diseases might fall on the spectrum. So for instance, sorry, that's my little guy in the background. Flu has a reproductive number of about 2. So that would tell you need to vaccinate about 50% of people if you weren't to get transmission anymore. Whereas smallpox has a higher reproductive number. So you can see that you need to vaccinate more people to achieve eradication. And smallpox, interestingly, is the only human disease we've ever successfully eradicated through global health campaigns. There is another disease, which is rinderpest, which is a bovine disease, but there are no other human diseases. We're getting close with polio, but we're not there yet. Measles has an even higher reproductive number, so you need to vaccinate a lot of the population in order to prevent transmission, and this is why vaccine refusal is so problematic, especially when you get clusters of pockets of large amounts of unvaccinated individuals, because then you can just get a lot of transmission. And then something like malaria has estimated to have a R0 of around 100 because it's spread by vectors. So one mosquito can consequently go on and infect lots of people. So if there were a vaccine, which there weren't, you would think you would have to really hit a large fraction of the population. So this is actually where the concept of herd immunity comes from, and I think that bears clarifying. So the idea is, what this model tells you is that you don't need to hit everyone in order to get rid of transmission. It tells you, depending on how infectious the disease is, you can vaccinate a certain amount of the population and then you can stop spread locally. And this is because you get rid of the wood or you get rid of the oxygen. An infected person just probabilistically won't contact a susceptible person anymore. And I think it bears mentioning that this concept is very different from what some people have been talking about in terms of, in my view, slightly changing the definition of herd immunity. If you're going to now say that just letting people get infected with a disease is going to achieve this, because the idea of vaccination doesn't rely on, needing to go through large death or human suffering in order to get to that point. And I think that that's a big distinction from sort of just saying something like, just let it run its course, let people get infected, when you're talking about vaccination versus natural infection. I think that's a very different story. So for instance, SARS-CoV-2, it's still not clear exactly what its reproductive number is. There are some estimates of around 2.5. So that would suggest you need to vaccinate about 60% of the population. So this sounds somewhat cut and dry at this point, but it turns out that vaccine immunity may not be lifelong. In fact, it's probably almost never lifelong, and neither may natural immunity. So the way we get immunity generally is through different types of adaptive immune responses. So your body encounters the antigen, and then through a number of different mechanisms, either by producing cellular immunity through memory T cells or humoral immunity through the production of antibodies, which are generated by B cells, you develop some amount to recognize the antigen if it meets it again, and then it lets you fight it off better subsequently. So because immunity is not necessarily perfect, another model is called what's known as the SIRS model. So now you eventually get to the recovered class once you recover from the infection, but if your immune response wanes, you're going to be transported back into the susceptible category. And for instance, influenza is modeled this way. There's also been papers modeling syphilis in this way. And this waning, as I said, could be due to your body losing its ability to fight the infection, or it could, for influenza, for instance, we know that it mutates all the time. It could also be possibly related to the fact that the antigen is just not the same anymore. And so you're just not good at recognizing it anymore. So there are some extreme versions of this model. This still looks pretty simple, but for instance, RSV, where we think there's some amount of maternal protection initially, and then there's the possibility for multiple reinfections. You can see that these models can get increasing complexity in order to capture the actual immune dynamics of what someone's immune response might be doing. And then, plugging that into your simulation for what dynamics will look quite different than if you just assumed that RSV were an SIR model type infection. So we wanted to use some of this to think about SARS-CoV-2, and in particular, we are curious about what the kinetics of the adaptive immune response to SARS-CoV-2 would do in terms of affecting the long-term predictions for what the dynamics of this infection might be. So SARS-CoV-2 is a part of the Coronaviridae family of enveloped positive strand RNA viruses. And a sort of phylogenetic picture would look something like this. So you can see that SARS-CoV-2 falls in the beta coronavirus genus. And what's interesting is that this genus also contains a number of viruses that cause common endemic infections in humans, like the common cold. And so a first approach, which others have thought to take it as well, was to say, well, since this is a new infection and we don't know what adaptive immune responses will look like yet, maybe we can look at these other related viruses and look at their adaptive immune responses in order to gain a little bit of intuition for how we might model SARS-CoV-2. So for instance, there's been data on how long antibodies wane for H-CoV OC43, which is one of the cold-causing viruses. And in general, the consensus is that, there's obviously variation between researchers and experiments, but a few months to a year seems to be sort of a generally accepted time frame. And this is consistent with the fact that we sort of repeatedly get cold infections with the same virus over time. In contrast, SARS-CoV-1, which was the cause of the original SARS, well, not the original SARS, the SARS pandemic earlier this millennium, antibodies in different studies were found to wane over the course of a couple of years, although it does bear mentioning that T cell memory was found to last for at least 11 years. So for SARS-CoV-2, you can imagine it doesn't feel like we're in the early stage of this infection because of the impact it's had on our lives, but we are. And so it's hard to really, we don't know what's going to happen in a year in terms of adaptive immunity. But there have been a lot of studies coming out trying to measure what this is going to be. So for instance, this was one study that found that already after 8 weeks after discharge from a hospital, there were decreases in antibody titers. And in particular, they varied between whether people were asymptomatic or symptomatic. But then a more recent study from Iceland found that there were persistent antibody levels for at least about 4 months. So we're still getting there in terms of measuring the kinetics of adaptive immune responses. But one important thing to point out is even if we could measure what the titers are, we don't know exactly what that means in terms of your protection. So that's known as a correlative protection. So just because you measure a certain number of antibodies in me, right now, I'm not able to tell you what that means in terms of my protection from a subsequent infection. So we took these ideas and we used what's known as an SIR and then in brackets S model. So it basically says we don't know that you're going to be fully susceptible to an infection, reinfection in the future. Let's try to think about it as more of a spectrum where you're not necessarily immune for life. You're not necessarily going fully back to the susceptible category, but you're somewhere in between. It's possible that you might be reinfected and that you'd be somewhat less susceptible or somewhat less able to transmit. So we're trying to bridge the gap between these two, and that's sort of hopefully going to allow us to cover all of our bases in terms of how the adaptive immune response to SARS-CoV-2 will pan out. So to do that, we rely on 3 parameters. The first is the relative susceptibility to secondary infection. The second is your relative transmissibility of secondary infections. And then delta would take to be the rate at which your immunity wanes. So to help ground this a little more, you can see that in the limit of not being susceptible at all to secondary infections, not being able to transmit secondary infections, and not having any immune waning, you just recover the normal SIR model where you get the infection once and then you're immune for life. On the other end of the spectrum, if you become completely susceptible just as much as you were the first time to secondary infections and transmitting secondary infections, and you have some positive rate of immune waning, then you're in just the SIRS model, which is what I showed you before, where you're protected for a certain amount of time and then you're just moving right back to the normal susceptible class. So our question was where, with this one model, we can cover these two ends of the spectrum just by setting the parameters in this way, but we can also cover regions in between. And just to show you what these sort of, very basic prototype expectations of what these models look like for dynamics for SIR, you see you get this one pandemic peak, and then the second later small peak is because I've included births, so I'm still fueling my susceptible class. But with SIRS, you see these future peaks happening sooner and more frequently because it's almost like you're supplying the susceptible supply faster because you get this initial pandemic, but then you get births plus people moving back to the susceptible class over time as their immunity is waning. So that's the model. I hope that that's somewhat clear. So I just want to talk about a couple of the results that we got. So the first one, so this is a bit of a complicated plot, so let me walk you through this. The X and Y axes here are your relative susceptibility to secondary infection. That's the X axis. So 0, as we said, was putting you back at an SIR model, whereas 1 is putting you at an SIRS model. And then your Y axis is how long your immunity is lasting. We assumed it's going to go between 0 and 2 years. So the top is showing you the time between the first 2 peaks that the model identifies, and the bottom one is showing you the size of the secondary peak. So this is normalized, so this is like fraction of the population. So in this, regions that are gray show that we didn't find a second peak within a period of 5 years. So this modeling was done assuming that the transmission rate has some non-constant component to it. So it looks something in general like this over time, whether that's due to climatic variation, whether it's due to people wearing masks and adopting non-pharmaceutical interventions, whether that's due to kids going back to school, possibly, as people have been saying, increasing transmission, or maybe just in winter, we're all going to be inside. Is that going to cause it? So we're not not necessarily claiming what's causing it. We're just saying assume that we have a non-constant transmission rate. Then what we find is this very sort of interesting non-intuitive result. So here I'm just plotting actual time series instead of these surface plots that I showed you before, where it's possible that when you have interactions with a non-constant transmission rate, better adaptive immune responses, which you can see by higher values of epsilon, actually can cause peaks to be earlier or larger. So basically what this is saying, first of all, you can note that the first peak is unaffected by anything that we assume about adaptive immunity, and this is consistent with what other researchers have found, but essentially it just says everyone is susceptible at the outset. So for the pandemic, it doesn't really matter what effect you assume climate is having or what effect you assume immunity is having. Everyone's susceptible, it's just going to run its course initially, but the longer term is going to be influenced by these more subtle factors that we're introducing with our model. But what we showed is that so because you have this interplay between when transmission rates are going up and possibly when the susceptible supply is biggest based on people moving back to the susceptible class from the recovered class, you can have sort of these counterintuitive results for the timing and size of subsequent peaks. When we turned off this non-monotonicity in transmission rate, so we just assume we have a constant value over time, you see that we get sort of the more intuitive result that as adaptive immune responses are worse or less protective, for instance, you can see that in the plot up here, as epsilon gets smaller or the duration of immunity gets smaller, then epidemics or peaks will become more close together and larger. Okay, so that's sort of the results in a world without vaccination, where we're really just interested in thinking about how adaptive immune responses will affect dynamics. But then what we wanted to do is introduce the possible effect of vaccination. So here we just added a vaccinated class to this model that I showed before. And we assume that it's possible that the vaccine is not perfect. And in fact, like I was saying, that's probably a decent assumption to have vaccines. If they're good, what they do is they prevent us from retransmitting the infection and they prevent us from getting severe disease, but they're not necessarily completely protective from any cell in your body ever getting infected by the virus. So what we assumed is we said, okay, let's introduce a vaccine that has a certain duration of vaccinal immunity. And then we made these area plots to look at how the composition of society changes over time under different scenarios about what immune responses might look like. So I'm going to walk you through these area plots. The first is a world with no vaccination, just so I can make that comparison. So the gray area is people who are fully susceptible. So you can see at time 0, everyone is fully susceptible. So these bottom plots are area plots, and then the top plots are related to clinical aspects, which we've taken to be a prediction for the number of severe cases. So this is basically just a fraction of the number of infections over time. So what you can see is that once the first, and then another thing I should say is that the plot on the left assumes poor natural immune outcomes. So it assumes that you're less protected from future infections, either through your immune response waning more quickly or you being more susceptible to reinfection later on. Whereas the right plot assumes that we get robust adaptive immune responses to this virus. So first of all, you can see it really does affect predictions for the burden and timing of subsequent peaks, and also for the fraction of the population that has some immunity over time. So obviously in the case on the left, when you just have more infections, you see these purple regions grow much more quickly until after 5 years, let me also say that these are not expected to correspond to any particular location in time. They're more qualitative projection. Eventually, the fully susceptible class is quite small because of the number of outbreaks that we've seen. So we can take these worlds and now we can say, okay, what happens if we introduce vaccination to them? And so again, we're assuming that the left is a poor adaptive immune outcome also for vaccination. So we're assuming that vaccine immunity lasts for a shorter amount of time than the scenario on the right. We're assuming that we have good adaptive immune responses and good vaccine adaptive immune responses. And again, you can see that the scenarios are quite different. First of all, even an imperfect vaccine definitely contributes to reducing the burden of infection over time, but having a really good vaccine and robust immune responses can almost suppress future outbreaks and sort of dramatically change the landscape of cases. So I think what this really shows is that to date, we're not entirely sure what adaptive immune responses will look like. Up until this time, you could argue that it's mattered less, because when a pathogen emerges in a fully susceptible population, all we can really do initially is adopt non-pharmaceutical interventions to turn down transmission. We can wear masks, we can keep distance. But over time, as it starts to move towards becoming endemic in the population, then factors like, what does immunity to the virus look like? What role is climate playing? That's going to start to affect what the long-term dynamics are going to look like. And what this work showed is that depending on where we fall on the spectrum, we can predict that the incidence over time will look quite different. So additional complexities that we touch on a little bit in the work, but I'm not going to talk about today. We did introduce MPIs and thought about different strategies for relaxing them. We thought briefly about the effect of vaccine refusal, especially if that's connected with contact rates. We thought about what might happen if we have heterogeneity between individuals in adaptive immune responses. And I think that this is actually going to be an area that we're going to need to do a lot more work on, because we know that things like age and super spreading events are important in the transmission of this virus. So we're going to need to think more about how they affect the long-term dynamics in connection with adaptive immunity. And also infection severity seems to vary a lot between cases. And related to that, and I think this is how I'm going to bring you back now to the engineering side, it seems like depending on the severity of the infection, this might be tied with the immune response that you get. So for instance, the same paper that I talked about before showed that people who had asymptomatic infections, so maybe they had a lower viral load, or somehow they didn't get as productively infected by the virus, also had weaker adaptive immune responses. So this suggests some connection between something that the body's doing and the number of viruses that you're meeting and what your body generates in terms of protective immunity. There's also possibly an association between viral load and disease severity, which I think again ties back to this point before. So I think that this is really the area where I see an obvious connection for tying in aspects of bioengineering to transmission. And so let me just take a couple minutes to explain what I mean by that. So I've shown you already that we can say, climate might affect transmission rate, or public policies such as NPI adoption, or people going to school or crowding inside. But I want to direct my lab to think about how bioengineering tools might help us think about this too. So for instance, I told you that mucus plays a role in the infection process since it lines the respiratory tract. And we know already from studies with influenza and other viruses that it interacts with virus domains and can prevent them from reaching underlying cell layers. So one thing that I want to think about is how we can tune the dynamics of a viral infection by taking advantage of this binding and by thinking a little bit more carefully about how it occurs and designing biomaterials that might be capable to interact with viruses in this way. Also, we can then go on to connect this with within host disease models, because if our biomaterials are really able to protect underlying cell layers and to exclude viruses, then we could see that as affecting what the viral load in the body would be, which we can then use to infer something about transmission. So commonly an assumption made in disease modeling is that if I have tons of virus particles floating around, then probably if I cough, I'm going to transmit more than someone who has very few virus particles floating around. Whether or not that's true is debatable. So to do this, we're going to use something known as cross-scale models, which connect the picture within the host to what happens at the population level. So for instance, this has been done for influenza. And I think this is interesting because it's been shown that influenza viruses associated with upper respiratory tract infections are more transmissible, whereas ones associated with lower respiratory tract viruses are more associated with pathogenicity. And both of these factors will go on to affect the value of R0 or subsequent infections. So I think there's a lot of room for thinking about how biopolymers like mucin and also physiological aspects of, what does the respiratory tract system look like in these different places, and why is it causing the different pathophysiological outcomes that it is, to connect this to what's happening at the population level. So that's what I wanted to speak to you about today. And I'm just going to acknowledge my PhD group and my postdoc group, and this work was collaborative with everyone, in particular my PhD advisors, Gareth and Katerina, and my postdoc advisor, Brian. And at this point, I'm happy to take any questions and also just shamelessly advertise for my new lab and anyone who might be interested in doing some research.
Thank you so much, Caroline. It was a sublime lecture. Really crystal clear, amazing. Kept me on the edge of the chair. So let me look in the chat box. Oh, okay. So is that all right, everybody, if I'll read the question on your behalf? If not, just shout, but I think I will go ahead. So the question, okay, Molly?
Sorry, I was just like waiting in line because there's already chat questions. So I raise my hand so that I can ask later.
Okay. No, just type in and we will read them all because we apparently have plenty of time.
Okay, sounds good.
Okay.
So sorry to cut in, but before we begin, does anyone object to their questions being recorded and being answered? No? Okay, thank you.
All right, so question 1. Is it possible to tune the physical properties of mucin gels after cross-linking via altering external environments? For example, changing environmental pH.
After cross-linking, you mean after you try to get them to form additional cross-links the first time, or after they cross-link with something else?
I guess once you form the stable crosslinks,
yeah,
but because the labile crosslinks are dependent on the pH, right?
Yeah, that's an interesting question. I think the stable cross, like the ones that are called like quasi-dynamic that cause them to aggregate into really long molecules, those are sort of done deals. There are therapies for CF and other conditions that rely on you just going in and like ablating those to break them up into small little chunks. But once you do that, you're done. You're not going to get them to reform. If you're thinking more about the weak sort of physical crosslinks, then I think we did do experiments where we cross-linked them by dropping the pH and then we changed the pH subsequent to that. I think it's doable to a certain extent. You know, it was tough when we were adding surfactant because then you can't really go in and take out the surfactant. But there is, like, this really is somewhat of a phase separation phenomena. So it's not exactly the same as just turning a cross-link on or off. It is sort of the whole conformation of the polymer changing. So I'm going to say maybe to answer that. My long answer would be maybe. Would probably depend.
Okay, thank you. Let's move ahead. What do you think about models other than compartmental model on COVID virus?
Are you talking about like machine learning models or agent-based models, or is there one in particular that you're interested in?
Well, allow me to unmute the person asking, or maybe they can unmute themselves and specify.
Yep, thank you very much. I was talking about agent-based, but machine learning models on COVID, I've never heard, so maybe that too.
Well, yeah, so I mean, I'm all for them. Obviously, they're more complex than a few ODEs. It really depends on what you're interested in looking at. When you want to look at things really at the level of individuals and exactly what they're doing, then an agent-based might be more appropriate, but obviously it's going to come at the cost of being more computationally expensive. So an agent-based model, for people who don't know, is essentially keeping track of every single person in during an infection process, and then saying, you know, modeling exactly what they're doing, and then having people transition to different categories. The advantage would be that you could introduce heterogeneity more easily by having some people interact and some people not interact. And these have been applied. So again, I think it depends on the scale that you're interested in and the phenomena that you're interested in. What I mean by machine learning models is more sort of just predictive, I don't want to use the word black box, but kind of black box models where you're interested in making a prediction without introducing sort of the physics of the underlying process. And those have also been somewhat successful. I think, again, it would depend on the data you have and what you're interested in predicting at the end of the day.
Thanks.
Okay, great. So then next question is, well, first, thank you for the interesting talk. Is the method you applied to tag mucin structure changes under pH change unique? Sorry, I misread. Is it unique to mucin structures, or could you apply a similar method to other scaffolds, such as collagen gels, to observe shifts in structural heterogeneity over time? I may have missed it, but
Totally. Yeah. pH is like a very common axis for gels, for biological gels. In fact, I used to be quite jealous of the people who studied things like actin, or more sort of classically understood biological polymers, where, if I remember correctly, I think it was much more understood what additional cross-linking you were getting if you changed the pH in that system. So this is definitely, and even things like scaffolds, drug scaffolds rely on this to a certain extent, as the pH changes as you move from swallowing it into your digestive system, it'll start to degrade, and then the drug molecules will be released. So this is totally a very common technique in biological gel design.
lovely. Then a question from Professor Kearney. Can data about COVID be used to identify model parameters or structure?
And data from COVID. Yeah, I think so. So this isn't, yeah, so I mean, data, population level data would be one thing, like case data over time. Is that what, I guess that's what you mean? But if that is what you mean, then I would say yes. And there are numerical ways to take the cases that you get over time and then to back out what susceptibility might be. Although these do depend on you having somewhat of an understanding of whether you have something like an SIR process or an SIRS process. But further, the data that we desperately need and that we're relying on is the animal models and the human data on adaptive immune responses over time, and that's totally going to help us to make better population level models once we understand what this immunity means a little better.
Yeah, I guess the other question is are there natural experiments being done that could help identify things? Are there ways essentially to manipulate either the input or the parameters of what goes on and see what happens?
To match it to what we observe. I suspect the answer is yes. I guess if I were approaching this problem, the one thing I would just worry about is the time series that we have. So it helps generally in epidemiological work to have years of data in order to really be able to confidently make predictions about the model that you're using. And if you only have a few months at best, it might be tricky, but you could just be careful with your statistics and your confidence intervals. And that would probably, I'm sure people are doing that, although I don't have the papers to point you to.
And I would guess that one of the problems also is your data is pretty noisy.
Yeah, probably, especially depending on where you're looking. Like a big city might be different from a small city. And then, of course, the big problem with COVID, which I'm not talking about here, but for all the people in my class, it's going to be a bit of a repeat, my COVID lecture, but you will get some new stuff, I promise. But one issue has just been calculating these numerators and denominators. So how many people are actually susceptible and how many people are actually infected. At first, before we had a decent number of tests, we didn't know what the infection rate really was at all. So I think that's going to complicate the model predictions too.
Thank you. Very nice talk.
Okay, for the next question, I'm going to unmute Molly because I'm afraid that I will mess up the emphasis in this one question.
I'm sorry, I will also show my face. Oh, hi, thank you so much for the talk. So I have two quick questions. The first one is regarding the choice of mucin variant that you use. I double checked with your paper. I think you used Muc5AC. And maybe I missed this part, but I was wondering why was that the choice of the sole mucin component used? Because to my understanding, there are many different types of mucin variants, both secreted and the cell-tethered form, and they're actually found in it. The variants that you see really depends on the location as well as the composition of the mucus. So, what was the reasoning behind the selection?
100%, you're absolutely right. There's, I think, I want to say order 12 different types of mucins. or 20. So we worked with 3 different mucin systems, and we got these respectively by getting pig stomachs, which let us get MUC5AC, and then pig intestines let us get MUC2, I believe. And then we also got MUC5B from us. We would sit there and salivate into a large container, and then we would purify the MUC5B out of our saliva. It was a very long process. So you're right. So, and we did these experiments with all 3 of those gels. We were particularly interested in the non-cell tethered mucins. So you're right, we didn't do experiments with the cell tethered ones. But I think the reason why we were particularly interested in MUC5AC for this study was because of the fact that it is in so many different parts of the body, and it makes up these gels with such different properties. So we are really interested in playing with that aspect. But I should note that, the results aren't exactly the same depending on which mucin you use, but at least between MUC2, MUC5B, and MUC5AC, there were similarities in the responses when we applied these perturbations to the gels.
I see. And my second very quick question is regarding the pH that you were, I think in the very beginning was the mucin man. You talked about how mucin can be used to protect the epithelial from harsh environment or from pathogens. And one of the example you used was pH 2 versus pH 7, and both pH were used in your experiment. However, I was wondering if the mucus or the mucin will experience a gradient of pH rather than just a constant level, because I'm not an expert on mucus, but from what I remember, the variety of mucin changes as you go from epithelial to the distal side. And I would assume that if you're closer to the epithelial, it will be more neutral pH at 7. Well, if you're at the barrier, it will be, a lot lower at pH 2.
Yeah. I mean, I think you might be able to tell me I'm wrong here, but I think that there is MUC5AC throughout the entire barrier. But you're right that the way that we move from 2 to 7 is by essentially the mucin acts as a buffer to buffer hydrogen ions. And so you establish a gradient that lets you go from pH 2 in the stomach acid to pH 7 at the cell wall. So you're right that you would expect that you would cover a span of pHs from 2 to 7 in this gel. So we did the experiments at pH 2, pH 4, at pH 7, but we focus on pH 2 versus pH 7 just because of pH 4 sort of always seemed to be a little bit of like neutral ground to a certain extent. But you're right, in general, I think you're totally correct that there would be a gradient that would be set up, and we could test at every pH. And there are experiments that have been done at pH 1, pH 6, so people do that.
I see, thank you.
Yeah, thanks for your question.
Actually, I like unmuting people. So, that's Danny. I'm unmuting, so please ask a question.
Yeah, thank you so much for your presentation. So my question might not be exactly related to your work, but I was wondering more specifically about how you model the transmission rate of diseases, particularly for COVID. So how would you model people wearing masks properly, or those that only wear it over their mouths, for example? Because we've recently seen a lot of emphasis on, wearing masks properly, and I was just wondering how you would include that in a model.
I think it's going to be a disappointing answer for you. We literally just put a prefactor in front of beta that you can decrease. So you could imagine that mask wearing reduces your transmission by a certain proportion. So you could just multiply by beta by some value between 0 and 1, and you can assume that that's the effectiveness of NPI adoption. We tried doing it for a different range of NPIs, and we also tried, like immediately relaxing NPIs versus gradually relaxing NPIs, which I think is more consistent with some of the stuff that we've seen in different places. But really it's just by, so I said that beta was basically the product of how many people you meet and how many of those meetings result in transmission. And so really you can just think of mask wearing or distancing as reducing one of those 2 factors. And so we just took it to be a proportional decrease in beta.
Okay, I see. Thank you.
Great. So I will keep on muting. And next question from Marzier.
Okay, I should say that I wonder if you use the same model but different parameters for different geographical locations. or is it better to use totally different models?
Yeah, that's a great question. I think the model itself would be applicable, but I think that you would have to adjust, things like your demographic inputs, your periods of when NPIs were adopted, potential influences of climate. I think you would just have to tune. I think that the nice thing about this model is how general it is. You can choose any value for these parameters that you want, but you'd have to tune it for your environment. So actually, we made a little interactive widget that goes along with this paper that you can find in the paper, and you can change. Say you can use New York City climate versus somewhere like Jakarta, which has much less variation in climate over the years. So you could pretend that that affects beta less if we think the climate is important. You can change how long you've adopted NPIs for. And so I think these are the real subtleties that you would have. Like, we don't mean this to be quantitative, but if you wanted to try to get more quantitative, I think you'd really have to adapt this to the specific area that you're dealing with. And I think in some ways that's why it has made, everyone, at one point everyone was like, what's the effect of opening schools? Tell me. But everyone was doing such different things in terms of NPI adoption, in terms of when the outbreak had started, that it was really hard to parse out the effect of just one thing when you're dealing with so many variables. And that's why you really have to tailor it to where you want to talk to, if you talk about, if you want to be quantitative at all.
Mm-hmm. Very interesting, thank you.
Okay. Maria?
Hi, I was wondering about the gels. Do you only use it for modeling purposes to study the virus, or is there any way that this can be introduced to our body to help us?
Yeah, that's a great area, and that's something that I'd like to do a little more of as I start my time at McGill. So some people, that one hot topic is to make what's known as like a mucin mimetic biomaterial. So it would be a synthetic polymer, but it's based on principles of mucins and how they form gels in the body. And so some people have made, sort of quasi-dynamic gels that, if you change the pH or you do this or that, they change their cross-linking and they change their properties. And I think there was at least one study that I read where, so I didn't get to talk about it today, but we did some work on cervical mucus barriers, because it turns out that preterm birth is associated in some cases with dysregulation of the mucosal plug, which essentially keeps the sterile uterus environment from the colonized vaginal compartment. And so something about the physicochemical properties of that plug changing can allow pathogens to go through, which could potentially trigger an early delivery. So we worked on sort of the transport side of that. We looked at diffusion of particles and small peptides through cervical mucus samples, but other people have started to think a bit more from an engineering design strategy of, say, designing a mimetic, a synthetic material that behaves somewhat like mucus, that you could, say, add to an area where there's mucus that's not doing exactly the job that it should, in order to enhance the protective role that it should be playing. So things like that are definitely a hot area of work. I don't know if this is complete science fiction at this point, or if it'll ever be possible, but to me, if we could get more stuff in the respiratory system that could, exclude pathogens better, then maybe that would be a way to prevent infection too, or whether that's not possible and we'll just deploy polymers everywhere that can bind virus, like on all the surfaces that we might encounter or something. So I think that there's a lot of room for making these deployed, as opposed to just thinking about their function in the gel. And that's where my motivation really is coming from.
Thank you, it's very interesting.
Okay, so I will voice the next question from Deepthi. Thank you for your talk. Can we introduce antigens against COVID without altering interactive properties of antigens? Will the cross-linking steps due to different pH affect antigens entrapped in the mucus?
That's a good question. I don't think I'm going to be able to give you a definitive answer on that, because I'm not sure exactly what kind of antigens we'd be talking about here. But so do you mean like something that we would inhale that would prevent, that would act against COVID, or where are you thinking with this? You can unmute TV.
Okay. I would like to know, like, if we say that antigens, there are few, a large number of antigens are available. We don't know which is. One second. Hello, can you hear me?
Yeah.
Yeah. I don't know like which antigen exactly, but I just want to know the viability of antigen will be remained after the cross-linking process inside the mucus, because for the cross-linking of the mucus particle, it may undergo different pH solutions. So I just want to know, like, if we entrap some antigens, so that if the mucus is able to carry those antigens inside the body, so it can interact with the COVID virus particles and taken away from the body without causing any problems. So I just wondering, like, the viability of antigens will be remained after the cross-linking of the mucus particles.
I mean, my guess would be yes, because the body has, like, there are a lot of antibodies in mucosal layers. There are antimicrobial peptides. There are things whose job is specifically to interact with pathogens and target them for immune clearance already in mucosal barriers. Also, I would think that this would be occurring in the respiratory tract where things are at pH 7. There's not too much worry about phase separation or anything like that. So my very naive guess, without having more information about the actual system that we're dealing with, would be to say that I don't see any reason why it would be hugely problematic, and hopefully that would be possible.
Thank you. Thank you.
Okay, now I'm unmuting Jesse.
Sure. First of all, thank you very much for a very interesting seminar. So my question's on the spectrum of severity and duration of COVID. So we're seeing a lot of different types of cases and outcomes, asymptomatic, mild, severe, or lingering. I'm wondering if all these types of outcomes would necessitate different models, or is it appropriate that we're baking them all into one model?
Yeah, so I think the extreme would just be to say, build an agent-based model, have each agent have a different length of duration and transmission, and see what happens in that fully individual scenario. But then I would say, short of doing that, there is some room for these simple models and thinking about things like that. And one particular way that we did that, which is sort of in the supplement of the paper, so I didn't go through it, was we sort of assumed that we had two groups within the population, and our motivation was to try to model like an elderly population that possibly got more severe infections, but then also potentially stronger immune responses versus a fraction of the population that maybe had a different type of immune response, and maybe a different like severity or duration of infection. And so we saw that to first order, if you don't introduce a massive amount of heterogeneity, our qualitative ideas are conserved. But I think that this is something that is going to be important to think about. And I think it's, it's undeniably important because we're seeing super spreading being so important for the transmission of this disease. And this model, for instance, doesn't tell you anything about super spreading. There are ways to introduce it by introducing something like, by changing the distribution of who transmits at what rate. And that can change your dynamics pretty substantially. So that the person who's really pioneered that work is named Jamie Lloyd-Smith. He has some really fundamental stuff about super spreading. So there are ways to incorporate that into ODE type models. But I think that you're absolutely right. And we're actively in the process of trying to incorporate a little more about variation in cases in the predictions that the model puts out.
Thank you.
Hi, thanks for the talk. I was just wondering about how or if R0 is relevant to a vector-borne disease.
Yeah, you're right. It's a little subtle, but there are, so the way that you can derive an effective R0 by sort of considering the SIR model in the mosquito population and the human population separately, and then basically lumping them together as far as new cases are concerned to back out an R0. So I think it is done, at least academically, but you're right that it sort of has a different meaning when you give an R0 for a vector-borne disease versus a person-person disease.
Thanks.
Should I adjust the gels by reducing the disulfide bronze? There was one, sorry, Doc, you go ahead. Sorry, I was reading.
No, I think, well, thank you. I already read it, so I don't think I need to reread it again. So go ahead.
Oh, okay. Well, the question was, I showed that we were manipulating pH, surfactant, and other parameters, but I also showed disulfide bridges. And so the question is whether I reduced the disulfide bonds to affect the gels. The short answer is yes, I did at least one experiment where I did that, and it basically just causes everything to, it just destroys the network. I think it was called the nuclear solution in our group. So it was not, yeah.
It's quite efficient, but and it's not possible to tune that in some ratiometric manner.
So you're talking about not the salt bridges, but the end-to-end bonds.
Well, I'm not sure. I mean, it's not really. Yeah, I saw you had disulfide bridge, and my understanding was that they were quite important, the rheology of the gel. But so if you, I mean, depending on the ratio, the concentration of your reducing agent, that was not, you couldn't modulate that in any proportional manner. That's what you're telling me, basically.
Maybe we could have. I don't want to say for a fact that we couldn't have, but you're right that those were really important because one mucin subunit, that these can aggregate into on the orders of tens to 100 of molecules long, so they become huge once this aggregation process happens. So yeah, it's maybe if we had chosen different agents or put a lot less in, but from what we saw, this wasn't like an easily tunable axis that we could use without really compromising the integrity of the whole system.
Thank you.
All right, so thank you everybody for your questions. Thank you, Caroline, for the wonderful lecture, and see you next Friday.
Thank you.
Thank you.
Thank you.
Thanks, everyone. Have a great weekend.